Hello, hi everyone. I'm here with another video. In this video, I will be discussing the Karnataka State Eligibility Test for Assistant Professor. I'm discussing Life Science question paper, the 2014 part 3 question paper. In the previous video, I have discussed the 2014 KSET part 2 question paper. So let's discuss one by one in this video. In this video, I might be discussing around 70 questions. So let's discuss one by one. So read out the first question. Polytene chromosomes are found in. So where you come across these polytene chromosomes. The polytene chromosomes The polytene chromosomes will be found during the interphase stage of the cell division. These polytene chromosomes are mainly observed in Chironomus larva, Chironomus larva, and also seen in the Drosophila. So, in these species, you come across this polytene chromosome, and these polytene chromosomes are observed or found during the interphase stage of the cell division. So, C is the right answer for this question. Then read out the second question. The differentiated part of the chromosome which gets associated with spindle fibers during cell division is. So consider this as a chromosome and this is centromere. To this centromere will come, there is attachment of the chromatids. These are the chromatids. So this is a typical structure of a chromosome. The center part is the centromere. On the side of the centromere, you will come across a special protein material that is the that is the kinetochore. And this kinetochore, it helps in the attachment of spindle fibers. It helps in the attachment of spindle fiber during the cell division. So C is the right answer for this question. Then read out the third question. Tata boxes and Pribno boxes are components of. Here, Tata, its homology is Pribno boxes. In these Pribno boxes, the main component will be the promoters. So the promoters are the main component of this Tata boxes. So C is the right answer for this question. Then read out the next question. The segmentation genes in Drosophila are divided into three groups: gap, pair rule, and segment polarity based on their mutant phenotype. Which of the following sequence of genes expressed from early to late embryo is correct? So here the right answer for this question will be the B. So B option is the right answer. Why? Because hunchback, hunchback, it is one of the gap genes. So this one will be a gap gene. So whereas even stripped, it is one of the pair rule gene. Pair rule. Whereas Fusitarazo, it is also a pair rule. Whereas the last one, wingless, it is a polarity segment polarity gene. So B is the right answer for this one. In this one, in this type, the genes will get expressed from early to late embryo. So B is the right answer for this question. Then read out the fifth question. During urine formation, the filtration of blood at the glomerulus is. So for this one, during the urine formation, the filtration of blood at the glomerulus is a pressure dependent physical process. So C is the right answer for this question. Then read out the next question. A technique for visualizing chromosomes aberration using fluorescent labeled DNA probes which are hybridized to chromosomal DNA. So, which technique will be utilized for visualizing the chromosomal aberration using the fluorescent labeled DNA probes? So, this is one of the method, that is chromosome painting. So, this chromosome painting, through which we can do a visualization of our individual chromosomes in a metaphase or interphase cells, so that a correct identification of both the numerical and structural chromosomal aberration can be observed. So, C is the right answer for this question. Then, next question. When two mutants having the same phenotype were crossed, the progeny obtained showed a wild phenotype. Thus, the mutations are, that is allelic. So, during this allelic reaction or allelic mutation, where we can come across this type, even though you crossed two mutants having the same phenotype, but the progeny has showed a wild phenotype. So, C is the right answer for this question. Then, eighth one, read out the eighth question. In humans, the haploid number of chromosome is 23. By independent assortment, how many possible different gametes can be produced? That is D option. D option. Through independent assortment, we can do 23 to the 23 power. So, D is the right answer. Through this one, we can produce so many different gametes. Eighth, D is the right answer. Then read out the next question. A lethal autosomal dominant trait with complete penetrance has a population frequency of 1 is to 50,000. What is the rate of new mutation? That is the B is the right answer. 1 is to 1 by 1 lakh. So B is the right answer for this question. Then 10th one, the correct order of performing DNA profiling is. So DNA profiling is also called as a forensic technique. Forensic technique. So, what are the methods utilized during the forensic technique? So, in this forensic technique, we first do is DNA isolation, then we will do the PCR amplification, that is polymerase chain reaction. Through this polymerase chain reaction, we can get a multiple copies. After getting a multiple copies, we will do the electrophoresis. Electrophoresis, where the method where the DNA fragments will get separated according to their sizes. After that, we will do the southern blotting. Southern blotting is done for the DNA isolation, then autoradiography, then the analysis of the DNA pattern. So, A is the right answer for this DNA profiling or the forensic technique. A is the right answer. Then read out the next one. The 11th question. Uptake of naked DNA by a bacterium is. So that is a transformation. So it is a method through which a naked DNA can be uptaken by a bacterium. That is transformation. Then 12th one, which part of the chromosome is involved in aging? Aging. That is aging will be observed by the telomere actually. Here they have given chromocenter. Let's discuss here. Actually, the telomere is also a part of this one, where the aging can be absorbed based on the telomere, as the telomere will get degenerated. So 12th one, B, they have given, according to this one, a telomere can also be a part of this one. Then read out the next one. 13th one, name the techniques to identify cells involved in transcription. Cells involved in transcription. Northern blots, restriction digestion, PCR and restriction digestion, northern blots and in situ hybridization, and in situ hybridization and restriction digestion. So see, through this technique we can study the transcription process. So C is the right answer for this question. Then 14th one, the formation of the acrosome. So formation of acrosome is basically involves the maturation of lysosomal enzymes on the acrosome, that is the part of the sperm head. So where it involves the maturation of the lysosomal enzyme or the hydrolytic enzyme. So B is the right answer for this question. This is middle piece and this is a tail and this is head. So 15th one, the primary regulator of Leydig cell secretion is. So Leydig cell secretion, Leydig cells will be present in between the seminiferous tubule, that too in the interstitial, or this Leydig cells are also called as interstitial cell. This interstitial cell or Leydig cells, they will do the process in the secretion of the hormones. So it is under the control of this one, that is luteinizing hormone LH. So this will helps in the secretion. So next one, 16th one, during oxidative phosphorylation in mitochondria, synthesis of ATP occurs due to. So in oxidative phosphorylation, so consider this as a mitochondria. Consider these are the complexes which we observe in this Krebs cycle. So sorry, in a electron transport chain, during this electron transport chain, there is a formation of ATP takes place in the mitochondria through converting the adenosine diphosphate plus inorganic phosphate to ATP. So it is because of the electrochemical proton gradient. So it happens because of the electrochemical, sorry, this happens because of the electro because of the electrochemical proton gradient. So due to this, there is a formation of ATP by the enzyme ATPase. So this enzyme ATPase convert the ADP plus inorganic phosphate into a ATP through the oxisome, that is F0 F1 particles. F0 F1 particles. So 16B is the right answer. Then read out the next one. Next question. A copper is associated with which of the following mitochondrial enzyme? So copper It is associated with the enzyme that is cytochrome oxidase or cytochrome complexes. So this cytochrome, it is mainly included or it is mainly composed with the copper. So A is the right answer for this question. The next question, 18th question. Enzymes, vitamins and hormones are common in. So enzyme, vitamins and hormones are common in regulating metabolism. So in order to regulate metabolism like catabolism and anabolism, so it will be included with enzyme, vitamins and hormones. So D is the right answer for this question. Then BRCA1 and BRCA2 are involved in diagnosis of breast cancer. Breast cancer. So breast cancer is named by BRCA1 and 2. So it is mainly with the cancer, that is breast cancer. A is the right answer. Then carcinomas are tumors of. So carcinoma, it is a tumor found on the epithelial cell. So C is the right answer. Then replica plating. Sorry. Replica plating is used to detect. So this replica plating is used to detect the mutant colony in a bacterial colony. So A is the right answer for this question. Next question, that is 22. A female sex organ of Batrachospermum is called as. So here Batrachospermum, it is one of the algae or the alga. So what is the female sex organ? That is Carpagonium. Carpagonium is the right answer for this question. Then read out the next one, 23rd question. During the dark reaction of photosynthesis is cyanobacteria. That is in cyanobacteria. What happens? CO2 is reduced to organic compounds. So that is CO2 will be utilized and it will form a starch, or the food synthesis takes place during the dark reaction. That is B is the right answer for this question. Then 24th, the net gain of ATP molecules in glycolysis. In glycolysis, at the beginning of the cycle, that is in that is in consumption consumption step, that is five first steps of glycolysis, where the two molecules of ATP will be utilized, and in the payoff step of glycolysis again the two ATPs will get released. So B is the right answer for this question. Then 25th question. So 25th question. The ecological agent responsible for Ticca disease of groundnut is. So Ticca disease of groundnut. So Ticca disease of groundnut were observed. It is because of the Cercospora species. That is Cercospora personata. So it is the causative agent of this disease, that is Ticca disease in case of the groundnut. So A is the right answer for this question. Then next question, 26th question. 26th question. The chromosomes appear as beaded structure. So beaded on string, beaded on string, beaded on string. So where you will see this kind of structure, that is during the prophase 1 of meiosis in the leptoteny stage. During this leptoteny, we will come across a beads on string structure. So A is the right answer for this question. Leptoteny is the right answer for this question. Then 27th, which of the following bacteria convert nitrites to nitrates? So nitrite means NO2 to nitrate. That is NO3. So it is done by the bacteria, that is nitrobacter. So this nitrobacter convert NO2 into NO3, that is nitrite to nitrate. So C is the right answer for this question. Then 28th, the plant cell differs from the animal cell in the absence of. Endoplasmic reticulum will be seen in both the plants and animals. Ribosomes will be found in both the organisms. Mitochondria will be seen in both the plant as well as animals. Whereas centrioles, these centrioles only present in case of the animal cell. So D is the right answer for this question. So 29th question. According to Baltimore's system of classification, positive sense single according to Baltimore's system of classification, positive sense single stranded RNA virus belong to which of the following groups? So here they have given groups, but they are asking regarding the single stranded RNA. So group 4, here it belongs a single stranded RNA. Group 3 contains double stranded RNA. Group 2 contains single stranded DNA, whereas group 1 a double stranded DNA. So right answer for this question will be, that is A is the right answer. So A is the right answer for this question. Then read out the next question. Bacterial blight of paddy is caused by. Bacterial blight of paddy is caused by Xanthomonas oryzae. This is the organism which can cause a disease, that is bacterial blight of paddy. So A is the answer for this question. Then 31, the etiological agent of head smut of sorghum is. Head smut of sorghum, that is caused by the organism, that is Plesiotheca sorghai. So this will cause a disease, that is head smut of sorghum. So 31, D is the right answer. Then 32, the antigen is. Antigen, it is a foreign organism or a disease causing organism which can stimulate the production of antibodies. So for this one, D is the right one. It is a chemical or biological substance that stimulate the production of antibodies by the body's immune system. So D is the right answer for this question. Next question, a 33. A cancerous cells are destroyed by the following type of cell. So cancerous cells are destroyed by natural killer cells. These natural killer cells, they directly goes and kill the cancerous cell. So 33B is the right answer. Then 34, phagocytosis may be characterized by which of the following statement? Phagocytosis, it means a cell eating. Pinocytosis means cell drinking. Here they are asking regarding the phagocytosis. It involves the uptake of cellular debris in large endocytic vesicle. So B is the right answer for this question. One last question, that is, consider the following statement. Consider the following statement and its elements are. So here cut and paste transposons. So IS elements are cut and paste transposons. These are sequence of DNA in genome of prokaryotes. These sequence are parasites of host genome. So answer for this one will be all the three, 1, 2, 3 are correct. So 35D is the right answer. So in this video I have discussed around 35 questions. The rest 40 questions will be discussed in the next video. Thank you.